In this video, we will discuss about the spatial transcriptomes and single-cell RNA-seq data. So the results of this combined both techniques on the cell-cell interactions on pancreatic tumor cells microenvironment, pancreatic tumor microenvironment. In the previous video, we have discussed that how the macrophages plays a role towards lipid metabolism of the pancreatic cells. If you need to understand this topic, you can go through our previous videos. And so let us start our main topic. So in this case, from the single cell RNA-seq data, they use these 3000 variable genes, and in order to understand how they come up with these 3000 variable genes, you can refer to their supplemental figures based on the supplemental figures that are presented in this paper. And if you want to access this paper, you can go through the reference that has been provided in the description. So based on their 3000 variable genes that they have identified in the single cell RNA-seq data, they then do the principal component analysis plot to reduce dimensionality. And after this plot, they can see that the samples varied differently. So in order to further validate that, they did the envelope plot of the three different pancreatic samples. So for this experiment, they used tissues from three different pancreatic cancer patients. So in this case, they can find out that different tissues have different kind of heterogeneity, and these things they have validated using the standard deviation of this pancreatic cancer cells from this standard deviation of the pancreatic cancers of this pancreatic tissues. And after that, they did the spatial transcriptomes on all these three different pancreatic tissues that can be seen here. And in these spatial transcriptomes, they have highlighted the cells, or they did the spatial transcriptomes, and in order to identify the characteristics of the cells, they use single cell RNA-seq data, and they can find out different kind of cell populations that are mentioned here in all the three pancreatic tissue samples. And since in our previous video, or since from the previous results, they have made a point, or they find out that macrophages plays a role towards lipid metabolism of the cancer. So the panel E focuses on the macrophage spatial transcriptomes descriptions. Here they can see that macrophages are present across different part, and in the case similarly in the pancreatic samples 2 and pancreatic cancer 3s, the presence of macrophages are clearly evident. And however, in the pancreatic cancer 2, the intensity is low compared to the case of pancreatic cancer 1 and pancreatic cancer 3. So after identifying that macrophages are present within these pancreatic cancers sample tissues, they did their SCOET package analysis to find out the cell-cell interactions, and each circle represents each cell lineage and their interactions to each other. However, the maximum interactions that they find out from this analysis is that epithelial cells interacts with this M1 macrophages, and once they thereafter they did the heat plot to find out the cell-cell proportion correlations. That is, within each cell, what are the within each section, what are the populations, or what are the ratios of the different populations of the cells. And in this case, they have find out that when compared for this M1 macrophage and epithelial cells relations, it's negative. And this is also in line with the literatures where they have seen that epithelial cells, or cancerous cells, has a negative correlations with M1 macrophages, since M1 macrophages has anti-tumorigenic properties, whereas M2 macrophages has pro-tumorigenic properties. So that's why they find the M2 macrophages and epithelial cells or cancer cells correlations is in the positive sections, or is in the positive range, and these are the Pearson correlation range. So their heat plots are also in line with the information that is available in the literatures, that M1 macrophages has anti-tumorigenic properties and M2 macrophages has pro-tumorigenic properties. That is, their cell proportions M2 macro M2 macrophages and epithelial cells are high compared to the case of epithelial cells and M1 macrophages. So I hope you, and what does this mean is that in a particular area, if M1 macrophages are high, then epithelial cells are low. However, in a particular area, if M2 macrophages are high, then epithelial cells are also high compared to the previous case. So I hope you understood that how from their spatial transcriptome single cell RNA data, they reveals that interactions of the different cell clusters and which interactions are higher, and also they are able to reveal the reveal using the heat plot that M1 macrophages and epithelial cancer cells has anti-tumorigenic. M1 macrophages has anti-tumorigenic properties against the epithelial cells, and M2 macrophages has pro-tumorigenic properties against these epithelial cells or cancer cells. If you need to understand more about their experimentals uh, analysis in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.